morning and welcome everyone to the track one of 2021 Midwest Manure Summit. My name is Amber O'Brien and I'm the Ag Educator for Calumet County Extension, and I'll be your host for this track. As with the first session, please keep your microphones muted and videos off during the webinar. The mute and video controls are located at the bottom left-hand side of your screen. I'm going to share a quick word from our sponsors. And without our sponsors today, we wouldn't have been able to put this on.
The Dairy Business Association is a dynamic, difference-making organization that empowers our members to lead America's dairyland forward. We connect with our communities and policymakers through advocacy, collaboration, and continuous improvement. As the most effective lobbying organization for dairy farmers and business partners in Wisconsin, we ensure the interests of the dairy community are well represented in state and local government. We work to foster a healthy business climate for dairy farmers and businesses and serve as a dependable resource for members who face critical challenges. As a member, you're directly involved in our policy positions and lobbying efforts, and can benefit from the public relations, social media, education, networking, and sustainability tools we provide. With you and for you, we are building a trusted and vibrant dairy community that balances business, environmental, and social responsibilities. Join us today in moving dairy forward. Go to dairyforward.com.
I'd like to introduce our first speaker, Dr. Amy Schmidt. Dr. Amy Schmidt is an animal manure management specialist at the University of Nebraska-Lincoln in the Biological Systems Engineering Department. Her appointment includes 30% extension, 40% research, and 30% animal science. Her areas of research and professional interest include nutrients fate and transport, pathogen fate and transport, water quality, and manure and nutrient management. Dr. Schmidt received her PhD in biological engineering from Mississippi State University and is a licensed professional engineer in Mississippi. Dr. Schmidt's presentation is called I Am Responsible and So Are You, and she's going to be addressing agriculture's role in antimicrobial resistance.
Thank you, Amber. Well, thanks everyone for joining today. This might be a little bit outside of what you normally would hear about at a manure-focused conference. Antimicrobial resistance is kind of a growing issue that has received a lot of attention in the past several years, and a lot of that is focused on the agricultural industry because of the use of antibiotics for livestock production. What I want to talk about today is what is AMR and why should we care about it? I want to talk about the I Am Responsible project, and then I'll finish up with what role we can all play in addressing antimicrobial resistance. So what is AMR and why should we care about it? The abbreviation AMR stands for antimicrobial resistance. Now, when we talk about microbes, we're including bacteria, viruses, fungi, all different single-cell organisms that are not visible to the naked eye. We often use the term antimicrobial resistant and antibiotic resistant interchangeably. They're not really the same thing. We have within antimicrobial resistance, we're talking about antibiotic resistance, antiviral resistance, antifungal resistance, and so on. While you'll probably hear me use the term AMR throughout the presentation, really the focus and the biggest concern within antimicrobial resistance is antibiotic resistance. So I want to share a short video with you, and this is for a website that the I Am Responsible team is developing. And so I will tell you that this is not the finished product, but it's getting very close, and so forgive the voiceover in it. It's not a professional voiceover, but this is a good 2-minute introduction to the project and why we should care about antimicrobial resistance.
This year, hundreds of thousands of people will die from an antimicrobial resistant infection. That number is likely to rise in the future. AMR is one of the most serious public health threats of the 21st century. Germs, bacteria, viruses, and fungi are everywhere. Bacteria are on and in our bodies, in the air, soil, water, and on plants and animals. Most bacteria are harmless. Many are even necessary. Some bacteria help us digest food, stay healthy, and produce foods like yogurt, cheese, and kimchi. but others can make us sick. When that happens, we count on antibiotics to cure the infection. Since they were discovered, they've saved millions of lives. It's hard to believe there was a time when illnesses we now treat very easily, like pneumonia or an infected wound, were likely to cause death. The length and quality of our lives have certainly improved thanks to antibiotics. But did you know bacteria can become resistant to antibiotics? All living things want to survive and reproduce to carry on their species. This means adapting to changes in their environment that threaten survival. Animals have been known to change in color over many generations to make them less visible to predators. For bacteria, viruses, and fungi, figuring out a way to block the actions of a medicine that used to kill them can allow them to survive and reproduce. This is called being resistant to the antimicrobial medication. When these medications we rely on to cure infections no longer work, infections spread more easily and last longer, causing greater damage to our body than even death. This year, 700,000 people will die from an antimicrobial resistant infection. By 2050, there could be 10 million deaths per year. Antimicrobial resistance is scary, and it's time for everyone to learn how they can join the fight against this growing health threat. We all have a role.
Let's first talk about what causes bacteria to become resistant to antibiotics. And this is, very much a antibiotic resistance 101 kind of discussion, not a very detailed, but I didn't want to avoid talking about this part of it because then people are thinking like, how does this even happen? So when I give this presentation in person, normally everybody gets cookies and chocolate chips. You just have to look at those on the screen today, so I apologize for that. But let's think of antibiotic resistance from this perspective. So we have bacteria. In this picture, they're represented by chocolate chip cookies. So these bacteria can be pathogenic, kinds that cause disease and illness in people or in animals, or they could be non-pathogenic. So we all have E. coli in our bodies all the time. Every warm-blooded animal has E. coli in their intestines, but they don't make us sick. They're actually beneficial to us. So we have our bacteria sitting out here, pathogenic or not pathogenic, doesn't really matter at this point. And then we have the resistance genes. And so this is the genetic material that gives bacteria or other organisms certain features. So just like we have genetic material that either gives us straight hair or curly hair, brown eyes or blue eyes or green eyes, this genetic material that we're talking about with resistance is a specific material that when a bacteria or virus or other organism has that within their cell structure, they can resist the effects of certain medications. And so there's millions, probably thousands of genes that code or give a bacteria the ability to resist an antibiotic. And these genes can exist within the bacteria, which gives us what we have on the right-hand side of the screen here, gives us antibiotic-resistant bacteria. So our chocolate chip cookies with M&Ms in them gives us a bacteria that has resistance to one or more antibiotic drugs. So those genes, like I say, they can exist within a living organism, living bacteria, but the genetic material can also exist outside of that living that living bacteria. And so when we think about how bacteria go from being just a regular old bacteria to one that's resistant to antibiotics, there's two ways that this happens. One is called selective pressure. So just like humans and every other animal, when we encounter a stressor, something in our environment that threatens our well-being, we adapt. Now, humans may take us much longer to adapt physiologically because we only regenerate, new generation every 20 or 25 years. With bacteria, they produce new generations in a matter of minutes. So they grow very quickly. And if they encounter some environmental condition, whether it's low humidity, high temperature, some other organism in the environment, a predator or some competitor or something. As that bacteria continues to reproduce, they're mutating or making changes in their structure that helps them fight off whatever this threat is. And so once that bacteria forms a mutation that makes it more able to survive in that environment, the bacteria without that feature are dying off, whereas the one with that beneficial feature continues to multiply and create more bacteria with the same ability to resist whatever it is in the environment that's threatening them. So one is just by pressure from outside causes the change internally with that bacterial cell. The other is horizontal gene transfer. So I like to think of this like with trading cards. I'm a baseball fan. I have lots of baseball cards, trade baseball cards. So I think this is a kind of a simple way of looking at this. We have a bacterial cell that possesses genetic material that codes for resistance to one or more types of antibiotics. So he's kind of got this shield that blocks the effect of antibiotics. So if we think of that genetic material as a trading card, bacteria can trade that genetic material between cells. So if we make a second copy and I give that trading card to another bacterial cell, now we both have it. And both types of bacteria are resistant to the same antibiotic or types of antibiotics that the original one was resistant to. So environmental pressure or swapping, trading genetic material are the two ways that this happens. And that's obviously a simplified explanation of it, but I don't think we need to go beyond that in this discussion. So the question, when we think about this increasing concern about antimicrobial resistant bacteria in the environment and in the human population, the animal population, it's human nature to want to say who's to blame. And if you're in that conversation at all about antibiotic resistance, you probably hear one of two things. One, it's agriculture's fault. We use too many antibiotics in our livestock and we're just causing resistance to grow, and then it's those zoonotic diseases are getting into humans, and then we have resistant infections that can't be treated and so on. The other side of the coin is the medical field. We prescribe too many antibiotics. We maybe don't follow good enough hygiene practices in clinical and hospital settings. So bacteria that from one patient can get transferred to another patient. But the fact is there's a whole lot of things that go into whether or not antimicrobial resistance continues to grow as an issue or whether we kind of get it under control. And so the antimicrobial, the IAMR project is focused on this idea that we all have a role. So no more blame game. Everybody has something they can do related to antimicrobial resistance. So as we look at the logo here, this represents government policy, things that might impact how antibiotics are used and distributed and developed. So for instance, if policy doesn't allow for pharmaceutical companies to take an antibiotic drug from conception to production and use within a short enough amount of time, it costs that pharmaceutical company millions and billions of dollars to get through all of the research and the testing and the licensing and being allowed by the government to actually use that medication in a human or animal population. So not to say that that's wrong, but policy can drive how new antibiotics are produced, whether or not we're producing new antibiotics, and how those get from the manufacturer to the consumer. Government policy also impacts what we can use in livestock production, right? We had the Veterinary Feed Directive go into effect a couple of years ago, and that says we cannot use certain antibiotics in livestock production for promoting the growth of the animals because those certain drugs are super important to human medicine. So it doesn't mean we can't use antibiotics, but they've limited how they're used and how farmers can get access to them. The second thing we have here is the medication itself, and that sort of represents our role as consumers and how we use medication, whether we follow a prescription the way we're supposed to, whether we get antibiotics and then after 3 or 4 days of taking them, we feel better, so we quit taking them, even though it said to take the whole course. Do we throw them down the toilet, flush them down the toilet when there's leftover medications and get that medicine into the environment? Do we share antibiotics with other people? So then we also have, like I said, the livestock industry and also crop production industry. So antibiotics are used in livestock production, fish, shellfish production, crop production. And the thing we need to realize is that anytime antibiotics are used, not this idea of overusing or misusing antibiotics, but anytime antibiotics are used, they're going to create some selective pressure that could cause organisms to develop a resistance or to trade that resistance among the different organisms. So when you hear the discussion that agriculture overuses antibiotics or medicine is misusing or overprescribing antibiotics, it really, there's an idea that we need to be more, have better stewardship of our antibiotic use. But the fact is, no matter whether you use it once or 10 times, each individual use of antibiotics has the potential to promote the development of resistant bacteria. We talked about medicine being part of it as well. And then this final icon, this represents the environment. So antimicrobial resistance is not human created. It may be driven by some of our activities or a lot of our activities. But if you go back to your kind of grade school or high school science and think about where did antibiotics come from? Where did we find the first antibiotics? They came from the soil. So there's different antibiotic compounds that the bacteria are living in the soil, and there's another organism in there, whether it's a fungus or some other organism that is competing for the resources in that soil environment. And maybe that fungus decides, if I produce this chemical, it will kill the bacteria, and then I have access to the food and the resources around me. And so when penicillin was discovered, it was a mold that landed on the petri dish where bacteria was growing, and that mold actually produced penicillin, or the compound that we now call penicillin. And so we discovered, hey, these other organisms produce things that can kill bacteria, and then we learned how to use those for human medicine. So thinking about the environment, it doesn't really matter if people had ever been on this earth, there would be bacteria and there would be resistance to bacteria, because anytime bacteria are present and something is present to affect their growth, then they're going to develop resistance over time. So definitely not a man-made phenomenon, but many of the practices that we employ on a daily basis can impact how that development of resistance spreads or doesn't. So the I Am Responsible project, we started this about 3 years ago, and we have two main goals with it. One, and this is very much an extension-focused project. While several of us that are involved are doing research related to antimicrobial resistance and the different manure management practices or animal practices that impact it, everybody on this project kind of comes together with two goals. One is we want to understand or improve understanding among food producers and consumers about what is the AMR, why should I care about it, what are the related risks, and what practices do we know are effective against the further development of antibiotic resistance or the infection of humans and animals with a resistant bacteria. Once we've gotten that part, the improved understanding, then our next, our goal is to motivate adoption of behaviors that actually impact are potential risks related to antimicrobial resistant organisms and bacteria. And we're usually looking at this from a food safety risk or a general human health risk. So improve understanding and then motivate adoption of behaviors is what we are focused on. And this project is nationwide. We have a number of researchers from all different areas of the country. These are folks who have university appointments, either research and/or extension. And as you can see, we have dairy science, environmental science, food safety, vet medicine. We even have a medical humanities person who's kind of our moral compass that tells us how to communicate with consumers, because we think science should talk for itself. And people like Dr. Nixon say, you know, you really have to appeal to their senses. If you want people to understand this and to care about it, you need to make them understand why it's important. So we have a number of folks working in this area. And our goal is that whether we're talking to food producers, like I do, working with livestock producers, or whether we're talking with food consumers, like our food safety folks do, we want to be sharing the same messages with them, that everybody has a role, and here are the things you can do personally to affect antimicrobial resistance. So here's the important part. Where do we all fit in? I want to talk, like I said, we have two audiences. One is our food producers, the other is our food consumers. Everybody's a food consumer, right? Some of us more than others. Very small percentage of the consumer population are also producers of food. But when you have that responsibility of producing food to feed millions of people around the world, you also tend to be under the microscope of people who don't know what you're doing and don't know why you're doing it. And it's important for you to understand the practices that you can implement that impact human health and food safety related to food animal production. And of course, this is a manure-focused conference, right? So we're going to talk about manure management as producers. There's also the idea of antibiotic stewardship and animal husbandry. And I'll mention those a bit more, but really my area is on the manure management side. But I want to make sure we realize as consumers, our hygiene, washing your hands regularly so that you're not transferring pathogenic bacteria from surfaces you touch to your body. Food safety, simple ideas like cooking our meat to the correct internal temperature, washing our vegetables if we're going to eat those raw, keeping meat products, raw meat products and raw vegetable products separate in our food preparation environment. And then we also have a role related to antibiotic stewardship. Like I mentioned, taking a prescription the way it's prescribed, not sharing it, not saving some for later, not quitting that prescription before you get to the end of it. So the focus on manure kind of brings us down to this idea of how can we manage manure to avoid distributing pathogenic organisms or resistant organisms beyond our livestock production system. We know that manure contaminants, when manure is land applied, they're going to move in one of two ways. They're either going to move with liquid by runoff, or they're going to move with soil by erosion. And so thinking about, whether we're talking about nutrient management or management of chemicals or contaminants, inorganic fertilizer or manure, our goal is we want to keep these things out of our water. And one of the best ways to do that is to slow down the flow of water or erosion from the surface of a soil, reduce the incidence of erosion and runoff. And any of these water quality protection strategies that, we talk about best management practices or conservation practices in agriculture, if they work for nutrients, which is what we really focus most on in animal agriculture, the nutrient transport. But everything kind of moves in one of two ways, like I said, runoff or erosion. So our goals should be to protect the soil surface, reduce the runoff water volume and reduce the rate that water runs off of a field, create a barrier between our production area and water. And of course, we want to always deliver nutrients when, where, and how they're needed, whether that's organic nutrients from manure or inorganic nutrients from fertilizer product. So let's look at some of the practices that we consider conservation practices or best practices for managing our soil surface and manure. So we think about residue cover. This is a practice that has become more popular in the last decade or so. When we keep the surface of the soil covered with some residue, we keep that surface, we retain more moisture in the surface. We reduce soil erosion because with that soil surface covered, when we have the impact of raindrops, that's not displacing soil particles that can then be carried in the runoff. And when we're controlling that loss of soil particles from our field, we're also controlling the loss of anything that attaches to those soil particles. So phosphorus is very much associated with the soil particles. And if we're not allowing soil to run off of our field, we're not transporting phosphorus into surface water bodies. And again, any of these practices that we use for nutrients are also effective against different types of organisms and contaminants related to them. Another one is conservation buffers. So having some sort of barrier between your growing crop or your crop field and a water body that's off screen here. Anytime we have this sort of a barrier, it serves a couple of purposes. It slows down the runoff, and then it also filters that runoff. So it allows sediment to settle out, it allows liquid to infiltrate. And often when we're holding things like bacteria or organisms in that area where they aren't running into water, eventually the UV rays from the sun take care of killing those organisms. So we know that with conservation buffers, not only are we controlling nutrient loss from a field, we're really filtering out a lot of other contaminants that could be carried with that soil and water that are coming off of a field. Reduced tillage, again, kind of a newer practice in the last decade or so that's really gained popularity. So this serves a couple of purposes. Again, it leaves that residue on the surface that protects the soil surface from wind and water erosion, but it also helps the soil structure. And soil structure is very important to changing the way that soil infiltrates water or the way that we lose soil particles through erosion. So when we are using, when we're not doing tillage, when we are adding manure, organic matter to our soil surface, we're creating bacterial activity in that soil that produces chemicals that cause soil particles to bind together. And when we bind soil particles together, we're increasing the aggregate size of the soil. And you get to a point where if the aggregate is too large for water to carry it, when the water flows, it's not going to leave the field as erosion. So manure, just like these other practices, is a very positive, has a very positive influence on soil properties and therefore on the transport of nutrients and other contaminants from the surface of our fields to water bodies. Application timing is another one. So obviously it's better to apply manure when we don't expect rain for a couple of days than to apply manure right before rainfall. We know from plenty of different research projects that we have lower concentrations of contaminants in runoff from a field site the longer we go between application time and that runoff event. So again, these are all, very basic and common practices that you already know about and hopefully are already using. But the idea is that they also tie back not only to this nutrient transport, but to the transport of pathogens and different organisms from the field surface. So I told you we'd mention antibiotic stewardship again. And like I said, we have new regulations with the veterinary feed directive that limit how antibiotics can be used on the farm. But overall, the focus is just on judicious use of antibiotics. So treating as few animals as possible, treating for the shortest time possible, and using the smallest dose possible. And in human medicine, we see this move to using some sort of testing to confirm what bacteria it is we're trying to get rid of. So if a person goes into the hospital because they have sepsis or bacterial infection in their bloodstream, doctors are going to hit them hard with some combination of antibiotics that we know will kill just about anything. But at the same time, they're going to culture that bacteria and figure out like what exactly are we dealing with. And then when we know what bacteria we're dealing with, we can specify the treatment to that particular bacteria. The same sort of progress is being made in livestock production where, for instance, with mastitis in dairy cows, is this a contagious mastitis or is it a environmental form? So is it coming from other animals, or is it coming from surfaces where these animals lie? And once the veterinarian knows which of these sources they're trying to fight against, then they are more capable of prescribing the particular antibiotic to that animal that will actually fight that particular type of organism that's causing the infection. So using testing to confirm an infection source so that we are using the most appropriate treatment falls under this idea of antibiotic stewardship as well. Like I mentioned with the human population, one of the keys to preventing or to avoiding having to deal with an antimicrobial resistant infection is to prevent it altogether. So just like we should be washing our hands and keeping our raw and our cooked foods separate and things like that, the environment in which our animals live and the well-being that they experience in that production environment impacts their health. It impacts their exposure to different disease-causing organisms and their immune system and their ability to fight off some of these infections. So, taking care of animals, making sure that that we're using practices that minimize stress on the animals and maximize their well-being are ways that we can prevent infections in these animals. The other way we do this is through vaccinations. One of the things that I think is really important as folks within the agricultural community is that we know how to talk about what we're doing. People may not always listen, right? But it's good for us to be able to say, I know you're worried about antibiotic use in agriculture, and I totally get it. And from my perspective, I don't really like to treat disease. I would rather prevent it. And so one of the things we do is we use vaccinations to prevent diseases before they even occur. So thinking through the practices that you use and how you might explain those to a consumer audience that's not involved in agriculture, I think is a really important part to addressing some of these issues of placing blame, maybe more than is necessary, on agricultural production for this antimicrobial resistance issue. So I just want to wrap up with kind of some key messages here. So when resistance to treatment occurs among bacteria and other microorganisms, those organisms are more capable of causing diseases in humans and animals, and they're more difficult to treat, which means we're spending more money to treat them. We're potentially treating an animal or a person for some extended period of time and then losing that battle and resulting in the death of that person or that animal. So antimicrobial resistance is a big deal, and we need to start taking it more seriously, I think, as a global population. And once we understand that, then we need to be thinking about what are the behaviors that I can start using that will reduce the need for antibiotic treatment. Better hygiene, better living environment for the animals, managing manure in a way that I'm not discharging pathogenic organisms or resistant organisms to a water body where other animals and humans are potentially going to be in contact with them and could develop an infection. And so, like I said, when we're using best management practices for manure, and we're using the soil conservation practices that we all hear about from NRCS, you hear about it from Extension, and many of you probably have a lot of these different practices in place. But when we're using these to reduce runoff and erosion losses from soil that's received manure application, we're not only reducing nutrient loss, but we're reducing losses of other contaminants like antimicrobial resistant bacteria and genes. So I want to leave you with this idea. We all have a role. There may be blame placed among different audiences, but what we need to remember is that every single person has something they can do to contribute to addressing this issue of antimicrobial resistance, because we definitely want to see the next several generations of humans have access to antibiotics that actually treat infection. We don't want to go back to that pre-antibiotic era where simple infections couldn't be treated and became very serious. So I will leave you with that message. My contact information is here. I welcome you to send me an email if you have a question or a comment. We do have a pretty good social media presence with our I Am Responsible project on Twitter and Facebook. So I encourage you to look us up there. And with that, I will go ahead and wrap up and take any questions that you folks may have. So thank you.
Thank you, Dr. Amy. Great presentation. We did get a couple questions in the chat here, and I don't know if you want to pull up the chat to read them, but I will read them off to you so everyone can hear. Okay. We've got one from Trish Daughtry. Did resistance coincide with confinement livestock production, and are occurrences of resistance higher in areas with confined livestock production?
That is a fabulous question. No, it didn't coincide with confinement of livestock production. Like I said, whether we have people or livestock or any other animals living in an environment, resistance occurs. There's folks that have studied polar ice caps and found resistant bacteria and resistance genes in these ice formations from billions of years ago. Pristine environments like newly discovered caves, when they sample water in those environments or soil in those environments, they're finding resistance to several different types of antibiotics, but those antibiotics have never been present in that environment. So it's absolutely not related to beginning to use confinement systems. But just like when you have people living in closer quarters, there's more chance for disease to be passed. And so there's more need for using vaccinations or antibiotics to treat infections. So we do contribute to it anytime you or I or our animals are using antibiotics, but we are definitely not at fault as farmers for creating this issue. And then, very interesting question, are occurrences of resistance higher with confined livestock production? There's been quite a bit of research that has looked at raised without antibiotic production systems and organic production systems. And what has been found is that it doesn't actually matter if livestock receive antibiotics on a regular basis or not. They still carry resistant bacteria in their gut, and they still carry resistance genes in their GI tract. There may be some differences in the types of resistance that they carry, but what we have found is it really, if even a person who never ever takes antibiotics in their whole life has resistant organisms in their digestive tract. And it's because it's such a complicated process. A totally harmless bacteria that encounters another bacteria from wherever that exchanges that genetic material, and now we're carrying that resistance. There's natural resistance pools in the environment. There are hotspots of resistant organisms in different areas of population. For instance, one of them that I talk about with college students, because this kind of doesn't bother them until it comes down to this might affect me, is antimicrobial resistant gonorrhea. And that is a major infectious disease concern in areas of high college student density. And we've gotten to a point with this particular bacterial infection where there's basically one treatment left that works for it. About almost half of all gonorrhea infections are found to be resistant bacteria. And our options for treating it have, reduced substantially over the last several years. So this is an issue that, it crosses many different lifestyles and geographic regions and ways that we work and live. And so that's one of our big reasons why we're pushing this. We all have a role, because it is kind of frustrating to see blame placed on individual industries or populations when it's really it's a global issue that we all have an impact on.
Yeah, absolutely. Thanks for that. We've got one more, another follow-up question. Are antibiotics in feed being adjusted to use the correct antibiotic, such as a veterinarian would to do if called to diagnose?
Yeah, so the veterinary feed directive was aimed at ensuring that in order to, so you have a broad spectrum or a broad application of antibiotics where it's in the feed for every animal. This is where we kind of got the bad rap in agriculture for giving an antibiotic to prevent disease and help the animal grow faster. And then there's the therapeutic use, so treating an animal once they have an infection and only treating the animals that are ill. So the veterinary feed directive is really focused on getting us past the point of using antibiotics on a wide basis within a population to prevent disease or to improve the production of those animals. Now, an interesting thing to consider is that we use antibiotics prophylactically in humans too. So if you've ever had to have a surgery, you probably took a course of antibiotics before you ever went in for that surgery, just to help your body be prepared to fight anything it encounters during a surgery, right? Because you don't want an infection when you come out of a major surgery. So we use antibiotics in that same way, but we don't use them on a daily basis to help us just be healthier altogether. And I think that's the thing that's really gotten a lot of focus from critics of livestock production. So if a veterinarian is called to diagnose an individual animal or a group of animals, just like with humans, they're going to look at it and say, okay, it's mastitis, whatever. They know what antibiotics they use to treat that, and they're probably going to start giving that to the animal right away. But the way we're trying to see things move is not only do they start that antibiotic right then, but they actually culture that organism and find out exactly what it is, and then go back and maybe adjust that treatment to specifically fight that particular organism.
Yep, thank you for that response. And I used to work on a dairy farm, and that's exactly what we did to diagnose the mastitis. So, all right. Okay, I don't see any other questions coming in. Thank you very much for answering those questions. Thank you, Dr. Amy, very much for your presentation.
Thank you. Have a great day, guys.